So what Song just went through with you and Mui is now you have data in the CDM. And so we're going to start from the premise that data is in the CDM, not just your data set, but maybe an entire network of data set. And now we are researchers and we want to generate evidence from data that has been standardized. What I'm going to go through very briefly is how I want you to think about creating a good research question, motivated by what we did during the Sisyphus challenge, but more generally the framework that we're trying to argue for. And I'm going to hopefully inspire you to say, if I can come up with the right research question, all I got to do is build some cohorts, and then Chan is going to show you exactly how we think about building cohorts and what tools we have to build cohorts. So I discussed yesterday our journey from data to evidence, and I highlighted briefly that one of the challenges when we think about evidence is that there's different types of evidence. There's clinical characterization, which is descriptive analyses of disease natural history, treatment utilization, incidence rates. There's population level effect estimation, so causal inference questions about safety surveillance and comparative effectiveness. And there's patient level prediction questions, so applying machine learning algorithms to answer questions about precision medicine and disease interception. And it's really this triangulation of characterization, prediction, and estimation that we recognize each of those requires different tools, each of them require a different evaluation framework, but they are not distinct from the perspective of evidence needs, because very often if we're asking some broad clinical question, we actually may need to characterize the population we want to study, to then do the causal inference about the medical intervention, to then be able to do the prediction to say who is going to experience that event. And in fact, during our Sisyphus challenge, we identified a range of really important questions, including what Jack shared with you with the fluoroquinolone study, where Jack shared one of the slides. We focused a lot on effect estimation yesterday, but he showed that in fact the question was how do we understand how often aortic aneurysm happens in a fluoroquinolone exposed population? Then we talked about, do fluoroquinolones actually cause aortic aneurysm? And then what we didn't get to, just in interest of time, was can we figure out which patients are going to have that aortic aneurysm? So these are very complementary types of questions. Now, what we've recognized in our community at large is that while we can classify things, and now we understand there are three really distinct types of analyses. They require three different distinct sets of analytic tools and evaluation frameworks for generating them. But it still can be hard for people to actually specify a question in these categories because they still are pretty abstract. And so over the last several years, what we've tried to do is make this very straightforward by providing a series of templated questions to fill in. And I would assert that by and large, certainly well over 90% of the questions that I anticipate are the kinds of questions we want to answer in the OHDSI community can be answered with one of these templated questions. And so I'm going to take a minute to go through this. I see several of you taking pictures. And this is actually the exact table that we used at the start of the Sisyphus challenge to basically say, specify whatever question you want. Just make sure you specify it by filling in the blanks. So it'll take us a couple minutes to go through this. If we are thinking of characterization, we can think of a question like disease natural history. And that can be structured very concretely in the format, amongst patients who are diagnosed with some disease, what are the patient's characteristics from their medical history? And so all you need to do as a researcher is if you want to do disease natural history, you just need to fill in the blank of your favorite disease. So an example would be, say, amongst patients with rheumatoid arthritis, what are their demographics, prior conditions, medications, health service utilization behaviors? And doing a disease natural history study should be that doing the disease natural history part is standardized, regimented, consistent. The thing that's different is the input of which disease we actually want to study. Because inherently, if we're doing disease natural history, we always want demographics. We always want to understand prior conditions and concomitant medications and health service utilization behaviors. What's really fundamentally different is just the input of the disease that you want to go after. If you're interested in treatment utilization, we can think about, amongst patients who have whatever is your favorite disease, which treatments were patients exposed to amongst some list of available treatment options and in which sequence. And in fact, in OHDSI, we have a treatment pathways package that will let you answer a very specific question, like amongst patients with depression, which treatments are you exposed to amongst SSRIs or bupropion or esketamine, and in which sequence? And all you need to do is actually just provide the disease you're interested in and the list of treatments you're interested in, and the rest of the analysis can kind of take care of itself. If I shift to population level estimation, we want to be very clear about what that specific causal estimate it is that we're trying to produce. So we may be interested in the question, does exposure to some drug increase the risk of some outcome within some time horizon? So you've got 3 fill-in-the-blank things you need to do. Give me a target cohort, give me an outcome cohort, and give me a time at risk. If you can specify those 3 inputs, we can specify a proper question that we can now fully implement and parameterize in that way. We also do comparative effectiveness research where there you need to not just specify a target cohort, but you also need to specify a comparator cohort. And then you specify an outcome, whether that be an adverse event, which would be a safety issue, or maybe it's actually the effectiveness marker of the benefit. In all the contexts, you just have inputs there. When we think about prediction, we could also think about answering the question, for a given patient who's some new user of a drug, what's the probability that that patient is going to experience some outcome in some future time horizon? Okay. The thing that I want you to see, though, is that we've enumerated a large menu of studies. I would contend that much, much greater than 90% of studies fall within these buckets. If you specify your question as just some open-ended, I'll just write whatever I want, it can be hard to translate your description into an operational analysis. But on the converse, if you can follow this template, not only have you standardized the meaning of the question that you're going to ask, but then that's going to directly feed into all of our standardized analytics so that you'll know exactly what you're going to do to get those results. So just to motivate this and then to hand it over to Chan, when you're thinking about coming up with the right question, recognize that in the OHDSI community, we're going to be doing analysis across a network of databases. All databases have been standardized. Our goal is to not just use standardized data, as you learned this morning, but also to use standardized analytics, because our community has established scientific best practices for how to answer specific types of questions. So for example, that treatment pathway example, we've not only developed an approach to do treatment pathways, but we've used it in our community to generate impactful results for various different diseases. Now, the thing that's the linchpin between the data and the analytics, whether it be any of these analytics, incidence rates, comparative cohort design, self-controlled case series prediction, the linchpin, the connection between the data and the analytics are cohorts. Those templated inputs, when I say, tell me your favorite disease, tell me your specific exposure, these need to be formally represented entities that we refer to as cohorts. And a cohort definition is a specification to identify a set of persons who satisfy one or more criteria for a duration of time. And that's a formal, very concrete, explicit representation of an idea that we have formalized through standardized tools. That's what Chan's going to actually spend a bunch of time making sure you understand the theory and the practice of how to create these cohort definitions. But the beautiful thing is if you can get that one idea and understand how to build cohorts, the beautiful thing is that all of these different analytics, all you're doing is passing some combination of cohorts into our standardized analytics to then get the standardized output for each of these different questions. So I'm going to turn it over to Chan, but I want you to be thinking about, actually, I'm going to show one more thing, and then I'm going to turn over to Chan. Let me flip over to this. So in the Sisyphus challenge, just to give you examples of these templated questions, we asked the community, give us any questions you want, and we identified 4 questions of interest. You heard about the fluoroquinolone study from Jack, but there was also an ophthalmologist in the US who was interested in the question about anti-VEGF treatments in blinding diseases and whether they cause kidney failure, and was able to fill in that template simply by changing the target cohort being anti-VEGF, the outcome being kidney failure. We had a researcher from the UK who was interested in, amongst patients with psoriasis, does exposure to Skyrizi increase the risk of cerebrovascular events in patients while on treatment relative to other biologics? So a comparative safety question following that same template. And we had another researcher out of Saudi Arabia who asked a characterization question interested in the incidence of PML amongst patients with multiple sclerosis. So each of these were different questions. All of them followed this standardized rubric, and all of what the teams really need to do was build those cohorts to pass them in. But that enables the entire suite of everything you're going to learn today in terms of being able to define, design, implement, and execute an analysis, that entire pipeline to work. So with that, I'm going to turn it over to Chan, who's now going to show you the most important part, I'll assert, which is those cohort definitions. Getting that right will then lead into all of the rest of our analytics.